Welcome, everyone. Thank you for joining us for our presentation titled Lateral Flow Assay Development from Lab-Based Assay to Diagnostics Test Prototype, introducing Cytiva's Development Services Platform. It's my pleasure to introduce you to Lee Jenkins, Senior Product Leader, Cytiva. For a complete biography on our speaker, please visit the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them via the Q&A box, and our speaker will address your questions at the end of the presentation. Please join me in welcoming Lee Jenkins. I will now turn the presentation over to him.
Okay, hi. So thank you for the introduction. So as introduced, I'm Lee Jenkins, and I'm here to take you through accelerating development and a focus on Cytiva's development services platform. So the presentation today is going to go through these agenda points. So I'll start off with an overview of the common development challenges. I'll move into an overview of the Cytiva diagnostic services, covering what it includes and how we overcome development challenges. I'll go into a typical custom development process and close with a case studies and a quick Q&A. So if you've got any questions throughout the presentation, please feel free to come into the chat and I'll pick them up at the end of the call. So going into an overview of the common development challenges. So you can see on this slide that we've outlined 6 different concepts which we've had feedback are typical challenges for developers going through a new product launch. I'll spend a couple of minutes going through each one, talking about why it's important to yourselves going through the process, what typical constraints are, things you need to be aware of, but also a little hint on how they can be overcome and what we can help to provide within the service. So starting on the first aspect, which is the access to experienced scientists. So one of the things about lateral flows which attracts people to developing them as a product concept is that they are pretty much unrivaled amongst other types of rapid tests when it comes to the speed and ease of use. The fact that it can be used by people away from central labs, they can be used at point of care, or they can be used in a home-based setting. However, the ease of use can quite easily, give a little bit of a complex development because it's quite a highly informed decision-making process. You need to have careful consideration of different elements of the test, including the nitrocellulose, the pads, and other key components. And it's quite easy to underestimate that. So access to a key scientist who has done this before can really help accelerate your development at this stage. You also have the aspect of, someone who has produced these tests before. They are aware of the common risks and the setbacks. Their experience has taught them from previously. It helps them to speed up the iterative assay design process, and it can lead to a more optimal solution that, saves you having to repeat testing or repeat development at a later stage. The third point is that, experienced staff also have knowledge of what works. And that can be quite key because when you're developing a new platform, you have to start from complete scratch, but you might have a scientist who knows, what detection particles work well with what membranes and what pads, for example, and it can significantly reduce your development time as a result. So all of these are key to making sure that, you're getting through your development process as smooth as possible. Okay, so the next part I'm going to take you into is robustness of design. So under robustness of design, talking about the complexity in the previous slide, it's very important that you manage that complexity and that you've got a working product at the end of it that is substantially developed in order to go into the market as it's intended to. So you need to be able to understand issues from layperson use. So you're not going to have clinicians in all instances using these tests. So it has to be error-proof in some respects. It has to be able to work in different humidities and different temperatures, and it has to have a good stability over time because you want to have your product on the shelf for, for as long a time as possible. They also need to be developed, and this one's quite important, using the full breadth of a product specification. So typically during development, it's important that you get products from the extremes of a specification. So you'll have a product with a nominal or an average rating, but within the typical release, it might vary. And you need to test that to make sure you understand any variation stack within your product. So you don't want the product to fall over once it's in the field or at a later stage as a result of not enough rigorous testing. And it's important that you consider all these aspects during the design process. The third part is, the world we're all working in with regards to lateral flow test is typically a medical device framework. We're all working to ISO 13485, and as much as possible, you need to ensure that everybody throughout your process is working to the same standard. So if you can have someone further down your process, your development partners or your component suppliers who are qualified to the same standard, it can significantly reassure your risk process. You've got quality products, but also it can help with audit trails and traceability of design history files at a later stage. Okay. The next framework, which we're going to touch on, is the rapid prototyping of designs. And this is something, again, which as you're going through the process, it's important that you're able to test your ideas, that you're able to work out how successful your design is going to be as early as possible in the process before you start investing large amounts of time, of resource, of money in this process. So rapidly getting from that idea to a proof of principle or a proof of concept is vital for proving your idea to whether it's your stakeholders, whether it's your investors, whatever it is that's going to trigger that next milestone of investment in a project. Again, key considerations here is how do you work with different design formulations in parallel to avoid time and delays in the process? How do you optimize the test conditions so that they're repeatable and that you don't get, again, variation later in your process? How do you overcome and evaluate commercial viability in a simple, low-cost way? And how do you access equipment and skills needed to quickly develop prototypes? In development stage, you might have a competing priorities between trying to achieve all of these things, but at the same time, you don't want to invest in expensive equipment, or you don't want to hire scientists when you can't give a long-term commitment to how long they're going to be required by your project. So being able to get rapid prototyping and quickly prove your concepts is a key part of development. The next one is access to funding. So I touched on it a little bit in the last slide, how rapid prototyping quickly can help you access funding, but it's key in any development, trying to prove as early as possible in your project the concept is viable. It releases the next phase of funding, but also it reinforces your resources you do have. You make sure you spend them in the right places, and this can give you challenges and competing priorities because there's that tie-up of how do you manage budget constraints against the needs for proving design and generating prototypes at the same time. One potential way around this is to look at your development in bursts, where you can outsource trenches of work if needed up front, so that's a simple way of freeing up that investment without having to overcommit or overstretch resources, because you get a very precise package of work delivered. Okay, so two more points I've got here. The next one is about access to infrastructure. And you can see in the image on the slide, a large biodot reel-to-reel dispenser. And the key part around this is that making sure that when you're in your R&D process, it's key to get access to a working, fully kitted out diagnostic lab, because one of the critical challenges is making sure you go from a lab scale test or just a benchtop simple test that you've developed yourself, but then making sure it's replicable on a large industrial scale. Everybody wants that test, which is going to produce millions of sales, and you have to have a way of being able to produce your product on the same scale in order to achieve those sales. So going back to the funding and the infrastructure elements, there's obviously a consideration of how do you handle this? Can you purchase, can you lease equipment? Can you get access to a lab, which gives you access to all this equipment in a timely way at a low expense. It's going back to the, being able to prove your concept in a very very timely way so that you can move on with your project and get through your milestones as quick as possible. And then probably the most important one, last of all, is protection of IP. So, we've got on this slide that, IP is precious and the lifeblood of your business, and that can't be underestimated. Now, IP is everything to new startups, and being able to protect that design is everything. You got to make sure that everybody you're working with throughout the process and at all steps that you're holding onto that IP, that there's no loss or unnecessary sharing. So choosing a partner or choosing the right way to work on your concept, there's important considerations that you get that back at the end of the process. So that's why I'm going to go into the next part of the presentation, is going from these 6 concepts, taking you through a process where at Cytiva, we try and support you. We'll partner with companies to go from an idea to a point of care test. So the comment on here is, I have a lab-based test that could be optimized into a commercially available point of care test, but people don't know how to do that, or they don't know where to turn for advice or for support. We have this service which can help provide the path from an idea or a lab-based test to the approved rapid diagnostic test. And, looking at development, the challenges, the frustrations, the hurdles, we can work with you to overcome that in a simple way. We can use our experience, our infrastructure, the components we have, and the scale of Cytiva to help, get people through this process in a very quick and timely way. So, as I mentioned, our service is titled Cytiva Diagnostic Services. And essentially, it's a way for people to navigate between the development process. So it covers everything from networking, collaboration, and transparency. So you can see on this slide, we have essentially the service built up. It's 2 pillars on the left-hand side. Then there's 2 things on the right, which, going back to those considerations from earlier, are key for everybody. So the services, first of all, is looking at training and education. So for people who have this idea, or they have a biomarker, or they have a ELISA test, but they've never built a lateral flow, one of the key elements we can offer is training for people to help teach them the basics of the process. We can get them started on, introduction to lateral flow, teach them typical pitfalls and issues which they need to avoid so that they can make sure that their project goes smooth. And this can either be in a classroom, a on-site or online education. So we understand that with the current pandemic situation, that maybe classroom or face-to-face isn't possible for everybody at the moment. So we could use virtual approaches, or alternatively, we're happy to invite people where it's safe to do so to come to our lab in Germany, and we can have a hands-on classroom session. The sessions can be tailored around anything you want to know. So typically it'd be a couple of days session up to maybe 4 or 5 days, where we can look through processes. So teaching dispensing protocols, teaching how to use a beer core for SPR analysis to select your detection pairs, whether it's looking to use dispensers, whether it's looking to do your component selection, so which membranes to choose, what pads to choose, how to make sure that everything fits together, how to get rid of any non-specific binding. We can do all of this within the framework. And the last bullet point is looking at workshops. So if you just have a challenge with something you want help coaching to overcome, you can use our expertise to help coach you through that. It can be a hands-on, in-depth session where we can set you up so that you're ready for the future and you know what to do next time this issue appears. The second element of the service is the development expertise. So again, several different forms this could take. So it could be methods development. So we can help with you to create SOPs or to create, bring this rigor to your process, which was mentioned earlier. We can look at methods transfer. So we can help you go between a development size product into a manufacturing size project, or we can either help transfer you into a contract manufacturing organization. There's methods qualification, so we can help you with your validation and verification of your designs. Or it can even be shorter, little informal bits of work, look at troubleshooting. So if you've got non-specific binding, or if you've got low sensitivity or accuracy of your test, we can help optimize that. And then finally, it's outsourcing of development. So this is looking at the whole wing-to-wing of the process. So taking it from that very early stage right the way through to a working manufacturing scale test, which we hand back to you. Obviously a lot more comprehensive work, but again, all in partnership and all in open transparency of working together throughout the process. The other two parts we bring, the IP protection and transparency. So, we're happy to share everything we're doing. It's a discussion, it's a collaboration, and we can talk through step by step what's next in the process. In agreement on how we proceed for each part of the process. If you're able to access the site, we can give full access to see what we do and work together doing development. Or if it's virtual, we can provide all the protocols electronically and have regular video calls. But the important part of this is that, what I touched on earlier about the IP being so vital, all of the IP transfers back to the original owner. At Cytiva, we don't own any of this. You have everything back. There's no licensing, and it's a clean hand back of everything to do with our assay. Materials and infrastructure. So, you've seen a few images throughout the presentation where we have this working lab, but, we can use our equipment and our processes, our staff to do everything from stability trials, lot release testing, complaint management. So if you've got some complaints and you can't work through what's going wrong in the test, we can help with that, or even raw material inspection and developing incoming and outgoing QC tests. Okay, so to move on from that, this is covering a typical customization process and development process and how we can work together. So, product concept is, very early stage idea of what you want to achieve, going through to a custom design. If it's working with us, we can look at customization of membranes or pads. We can help work with any bespoke designs you want, whether it's a unique cartridge or a unique lateral flow test. Going across optimization and validation, so we can help with small scale prototypes and then taking that onto a larger scale. Manufacturing, so once you get your product through and onto the market, we will support you throughout that stage, and then obviously ongoing support. So touching on this, once you're in the field, what do you need? Is it a regular supplier product, or if you have any sort of performance issues, we can help coach you for that as well. So that's the presentation I have. I'm going to just run a short video to conclude the session, and then I'll jump back on to take any questions from the Q&A. So feel free to put anything in the chat, and I'll come back to you and speak to you shortly. Thanks, everybody.
My name is Atherton. I am the CEO of a biotech startup that is called Diagnostics. We are developing a disease diagnostic and surveillance platform where you put blood on the diagnostic, and in 10 minutes it tells you if the patient has Ebola or not. And then it sends a message to an online platform and tells us where the patient is located, and then predicts where the disease will spread. For the past 3 days, we've been doing a diagnostics development workshop in Lateral. Our experience with the coaching workshop has been truly educational. It is rare to be able to speak about the science, the production, and the business in one conversation without a pause. Because we have a conversation that moves from the technical bits to the manufacturing, to the business, to the intellectual property. Something that is rare to find in the past 3 to 4 years that we've been speaking to people about the work that we do. Being a lean startup means that we don't have a lot of the infrastructure and equipment that is present at this facility. But being able to work with the equipment and the facility has helped us to see what we can grow to become and to envision what our later stage R&D and scaling up will look like.
My name is Charlotte Masurumule. I am the principal scientist at Diagnostics. This experience has allowed us to gain a better perspective for where we want to be and what it takes to get there. It has allowed us to forecast potential challenges along the way, and has allowed us to refine and redefine our research and development strategy.
And so the different challenges we have been addressing have been on simplifying the interaction between our diagnostic and the patient and improving the impact of that. For the next 18 months, we will definitely change our course. The work that we've been doing here has helped us to reshape our R&D strategy and to become more efficient.
Okay, so the next step is getting back to Oxford, getting into the lab, and continuing with our R&D, and continuing to refine our product development pathway.
And make our product at a much more efficient pace. Because the lessons that we've been getting in this workshop have saved us months of development.
So I would definitely recommend that other companies enter this competition. The program has allowed us to be able to have a fuller, more broader perspective of R&D to manufacturing to distribution and beyond to the end user.
Thank you, Lee, for that outstanding presentation. We will now move into the live Q&A portion of the presentation. As a reminder, please submit your question via the Q&A box. So our first question is, can you give an example of the type of tissue this service can help with?
Thank you. So the example of a service that we've worked with previously is that we had one company who approached us to help with troubleshooting of the assay. So they market, but they were getting with accuracy of their results in the field. So we helped them identify what the root cause was, specifically in this case that during development they only tested components from the middle of the specification ranges across several items. And then as the product went into the field, there was issues with one of the items still being in specification, but being slightly away from the normal center of the range. And then this caused some variation they would have with the accuracy. So after helping identify the root cause, we could very quickly help to resolving the problem. So I think it's a perfect example of what we. Other examples are like the Hutano diagnostics video. You know, as a customer where we specifically took them from an idea into the feasibility of the concept.
Great. Okay, I've got another question for you. Will Cytiva own the IP of any element of the test post-development?
No, so hopefully, I tried to touch on this as part of the presentation, but I know this question came in early. No, so Cytiva as a company, we try to be as transparent as possible. So we make sure that all the agreements up front specify this, but we want all of the IP back to the diagnostic developer. Now, we are not a diagnostic company. Our interest in this is helping our customers get to the market as quick as possible. So the only IP we maintain is around manufacture and supply of our components. Otherwise, everything else goes back to the DX company for the regulatory filing of the test.
Okay, thank you. Third question, will Cytiva be able to help with the manufacture of my test post-development?
So hopefully the person asking us in the session earlier with John from Amazon Health, who was talking about contract manufacturing organizations. So at Cytiva, we can't help with the actual manufacture of the test normally. I encourage you to get in contact with us to discuss this. But what we can do is help with the process of transferring into a contract manufacturer. So we can help you go through the process to make sure that everything is developed, that you have all the correct protocols, that you have identified a CMO to work with, and then we can help you, during that process. If anyone wants any more advice or conversation around this, I encourage them to get in contact and we can discuss on a case-by-case basis.
Okay. Our next question. Is this service only available in Europe or is it worldwide?
So it's worldwide. So the resources for this are currently based in Germany. So in our manufacturing site in Dassel, which is where we produce our membranes, such as the nitrocellulose going into lateral products. But it's, yes, worldwide. So at present, we have the Germany base. We are looking into expanding into a China base for 2021, albeit that will be on a smaller scale, but yeah, open to working with any customers across all regions.
Great, thank you, Lee. Next question. Does Cytiva have loyalty to any specific reagent supplier as part of their development?
So, two answers, no. So we have suppliers we work with, generally based upon experience of what we feel provides a quality product, and people we know with stable supply chains and product readily available. But if a customer has any specific requirement, we can discuss this with them. So if they have a particular supplier they use in their test or people they don't want to use, obviously we're happy to consider as part of the scope of the project. The best approach for this would be for the customer to, again, get in contact with us, and we can talk details about what they want and understand, what their requirements are.
Okay, got another question. Does this service have to be performed at the Cytiva site, or can it take place at the customer site?
Interesting question, because given the pandemic, it's a slightly different slant on this question, I guess, to normal. So before the whole world changed due to COVID-19 and the current endemic, we were looking to take this as a service that we could visit the sites. Obviously, they would have to have a diagnostic lab and able to have the resources to host the sort of work involved. But we were also encouraging people to come visit us in Germany. But the change and how we're working now is that obviously we're not traveling as much and people aren't coming over to us. So we're working a lot more digitally and by electronic means, so a lot more video calls, a lot more Skype and Teams calls. So, happy to talk with a customer on what works best for them, but it'd be digital in the future. It could be either approach depending on what they have available. If they come to Germany, it obviously allows us to show the site and to help understand what manufacturing around the nitrocellulose products, etc. But case by case, happy to discuss with customers what works best for them.
Okay, thank you, Lee. Our next question. Are you looking at developing COVID-19 antigen lateral flow test?
Question everybody asks about this. So hopefully it's been clear in the presentation that we don't develop our own tests. When we talk about IP and about customers, this is all about taking their projects and helping them get onto the market quicker. So we're not developing our own COVID-19 antigen test, but we do have some active projects we are working on which are trying to do so. So we've had some press releases previously. Those are really available on the internet with a little look around. But yes, those are projects we're working on, and we're open to discussing anything that people want to work with us on.
Okay, our next question. What is the general timeline and cost from product concept stage to manufacturing scale?
So this question is very dependent on what the customer is trying to achieve. So what the test is, how many targets, whether it's a single or multiplex, in what stage of product concept they're at. So it is awkward to give a direct answer to this on the forum. Probably, the best way would be for us to contact the customer around this event, and we can discuss, directly what they're trying to achieve. But timelines, a typical diagnostic test, you would traditionally be looking 2 to 3 years to go through a whole development, get through trials, get around the market. Around COVID-19, we've been forced to look at a lot shorter term. So we're typically looking more than 6 months for the development. That's put a lot more urgency and pressure on the timelines. But encourage the customer. We can talk to them specifically about what they're doing, understand their test, and then a specific quote on what it would cost and the timelines involved.
Okay, looks like we have one more question here. What are some of the factors that need to be considered while transferring the manufacturing process from one facility to another?
So again, I mentioned earlier that there's a presentation earlier today from Appleton Health. This was specifically what the topic was about. So I can summarize quickly, but John spent 30 minutes plus a Q&A going in depth in this topic. So I encourage this to look at that webinar for more details. So essentially, it's all about ensuring that the test has the correct level of stability, so that it can go from a bench top scale. Your conjugates, once they're dried, they're able to withstand the transfer into a larger manufacturing process. That you have a robust design, so that it's all transferable and that it works and can handle.
Well, it looks like you may have frozen up, Lee. Can you still hear us? Okay, well, it looks like we've lost Lee's video. So any questions that are submitted in the on-demand period can be answered by the speaker via email. Thank you again, Lee, and thank you to the audience for your outstanding questions. This presentation will be available for on-demand viewing for 12 months. Please remember to share it with your colleagues who might be interested in today's topic. And don't miss out on the other presentations on our agenda. Visit the Agenda tab in the auditorium for a full listing. Thank you again for your participation. And until next time, goodbye.